So the regulation of gene expression. So regulation of gene expression refers to like at various level. Okay. Uh, that gene expression results in the formation of polypeptide. Okay. Now in eukaryotes, the regulation could be extended at transcriptional level. So formation of primary transcript, then processing level, regulation of splicing, transport of mRNA from nucleus to the cytoplasm, and at translation level. Okay. Now the genes in a cell are expressed to perform a particular function or a set of functions. If an enzyme called beta-galactosidase is synthesized by E. coli, it is used to catalyze the hydrolysis of a disaccharide, that is lactose into galactose and glucose. Okay, the bacteria use them as a source of energy. So if the bacteria do not have lactose around them to be utilized for energy, so they would no longer require the synthesis of the enzyme beta-galactosidase. Therefore, in simple terms, it is metabolical, physiological, and environmental conditions that regulate the expression of genes. The development and differentiation of embryo into adult organisms are also a result of the coordinated regulation of expression of several sets of genes. Now, in prokaryotes, control of the rate of transcriptional initiation is the predominant site for the control of gene regulation. So, in a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affect its ability to recognize start sites. So, these regulatory proteins can act both positively activators and negatively repressors. The accessibility of promoter regions of eukaryotic DNA is in many cases regulated by interaction of proteins with sequence termed as operators. It is regulated by interaction of proteins with sequence system as operators. The operator region is adjacent to the promoter elements. In most operands and in most cases, the sequence of the operator bind a repressor protein. Okay, each operon has a specific operator and specific repressor protein. Okay, that is so lac operon that we study. Okay, so the lac operon, the elucidation of lac operon was also a result of close union between a geneticist Francis Jacob and biochemist Jacob Monod. Okay, they were the first to elicit the transcriptional regulatory system. So in lac operon, lac refers to a lactose. Okay, lac refers to a lactose, and a polycystonic structural gene is regulated by common promoter and regulatory genes. So these lac operons are very common in bacteria, and it's operon. Okay, now it consists of one regulatory gene. Now the I gene is the regulatory gene. Here the term I does not refer to inducer, it is derived from the inhibitor. And three structural genes ZYA. I gene codes for this repressor of the lac operon, right? Now Z codes for beta-galactosidase, BGAL, and on Y codes for permease enzyme that increases permeability of the cell to beta-galactosidase, and a gene code encodes for transacetylase. Hence, all the three genes produced in lac operon are required for metabolism of lactose. So, as you can see, in the absence of inducers, so PPO ZYA. Okay. So you can see there is I regulatory gene, then promoter, and then operator. So repressor mRNA form, so repressor protein is formed, it binds in the absence of inducer, that is an inducers. Now inducers, like you see inducers. Now glucose or galactose cannot act as inducers for lac operon, and I told you, I'm going to tell, like in the presence of inducers such as lactose or allolactose, they act as inducers, right? So you say in the presence of repressor and in the absence of inducer, repressor binds with the operator region. So a repressor binds to the operator region and prevents RNA polymerase from transcribing the operon. Okay, so RNA polymerase could not bind to the promoter region, right, to start this transcription. Now in presence of inducers, inducers can be like allolactose or lactose. So inducer, it inactivates the repressor, and then when RNA polymerase binds to the promoter and transcribe the process. So then ZYA also acted, and it forms a like mRNA through the transcription, and then this like mRNA goes under the translation, and like it forms like protein enzymes like beta galactosidase, permease, and transacetylase. So this is the in presence of inducer, right? Now lactose is a substrate for the enzyme beta galactosidase, and it regulates switching on or off of the operon. And it regulates switching on or off of the operon. Hence it is termed as inducer. Okay. So in the absence of a preferred carbon source such as glucose, if lactose is provided in the medium of the bacteria, the lactose is transported into the cells through the action of permease. Remember, a very low level of expression of lac operon has to be present in the cell all the time, otherwise lactose cannot enter the cell. Okay. Remember, a very low level of expression of lac operon has to be present in the cell all the time, otherwise lactose cannot enter the cell. The lactose then induces the operon in the following manner. So repressor of the operon is synthesized all the time. Repressor of the operon. So repressor is synthesized all the time constituently from the IgG. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. Now in the presence of an inducer, okay, such as in the presence of an inducers such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. Essentially, regulation of lac operon can also be visualized as regulation of enzyme synthesis by substrates. So, and substrates is the lactose, right? Allolactose or lactose. Now, so the regulation of lac operon by repressor. Regulation of lac operon by repressor is referred to as negative regulation. So lac operon is under control of positive regulation as well. Okay, now the Human Genome Project. So Human Genome Project, right? So HGP, that is Human Genome Project, in 1990. So this sequencing of human genome was launched in the year 1990. Okay, so Human Genome Project was called a mega project. Okay, the Human Genome Project was a 30-year project coordinated by US Department of Energy and National Institute of Health. Okay, the Wellcome Trust of UK became a major partner. Japan, France, Germany, China, others also contributed. The project was completed in 203, started in 1990, 30-year project. By this project addresses the ethical, legal, social issues (LC) that may arise from the project. Now methodologies. Okay. Now the methodologies. So now the methodologies, right? The methods involve two major approaches. The methods involve two major approaches. So one approach focus on identifying all the genes that are expressed as RNA. So one approach focus on identifying all the genes that are expressed as RNA. Okay, referred to as express sequence tags, ESTs. Okay, identifying all the genes that are expressed as RNA referred to as express sequence tags, ESTs. Okay. Now the other method is blind approach of simply sequencing the whole set of genome that contain all the coding and non-coding sequence. Okay, that contain all the coding and non-coding sequence, the latter assigning different regions in the sequence with functions, and later assigning different regions in the sequence with functions. Okay, so it's termed as the sequence annotation. Now for sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes Okay, now the cloning resulted into amplification of each piece of DNA fragment so that it subsequently could be sequenced with each. Now the commonly used hosts were bacteria and yeast. The vectors were called bacterial artificial chromosome, that is back, bacterial artificial chromosome back, and yeast artificial chromosome, that is yak. So the fragments were sequenced using automated DNA sequencers that work on principle of a method developed by Friedrich Sanger. Okay, the Sanger also credited for like determination of amino acid sequences in proteins, right? Now this sequence were then arranged based on some overlapping regions present in them, right? Computers and all these things are needed. Now the sequence of chromosome 1 was completed only in May 206. Okay, this was the last of the 24 chromosomes, that is 22 autosomes and XY to be sequenced. Now okay, some repetitive DNA sequence known as some repetitive sequence DNA, these are like the microsatellites, right? The silent features of human genome. The human genome contains 3164.7 million base pairs. The human genome contains 3164.7 million base pairs. The average gene consists of like 3000 bases, with the largest human gene being the dystrophin, has 2.4 million bases, right? And average gene consists of like 300 bases. Now total number of genes is estimated. So almost all 99.99% nucleotide bases are exactly the same in all humans. Almost all nucleotide bases are exactly the same in all humans. Okay, the functions are unknown for over 50% of the discovered genes. 50% of the discovered genes. So less than 2% of the genome codes for proteins. Repeated sequences make up very large portion of the human genome. The repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamic and evolution. Chromosome 1 has most genes like 2968, and it was completed study in 206. 2968, and Y has the fewest, like 231. So scientists have identified about 1.4 million locations where single base DNA differences, that is SNPs, that is single nucleotide polymorphism. These are the SNPs. 1.4 million locations where single base DNA differences, right? SNPs, single nucleotide polymorphism, occurs in human. So this SGP can useful in like disease associated sequences and tracing human history. Applications and future challenges. So DNA fingerprinting. Okay, so as stated, like 99.9% of the basic among humans is the same, right? DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as repeated DNA, because in these sequences a small stretch of DNA is repeated many times. Now these repeated DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. So DNA fingerprinting involves identifying differences in